I'm going to go ahead and get started. Hi everyone. Uh, my name is Sujit. I'm an assistant professor here at Princeton in chemical and biological engineering. And it's a real pleasure and honor for me to get to give you this lecture as part of this summer school. So thank you to the organizers and to Josh for the invitation. And I also wanted to give a shout out to my students, Daniel, Jenna, and Tapa, who led a really fantastic discussion section this morning. And to all of you students who submitted questions, I thought your questions were incredible and really on point. And I hope I'll answer some of them throughout this talk and also just give you an idea of some of the things we've been doing. And if you have any questions or comments and want to follow up, please find my contact details and send me an email or any of my students. So I changed the title of this talk a little bit to something that I thought was a little more catchy and representative. I'll tell you about some of the work we've been doing in my group looking at how bacteria move in heterogeneous media. And in general, what we do in my lab is we study various kinds of dynamics in heterogeneous and porous media. So I'll use those two terms interchangeably. And in the discussion section, Tapa, I think, described what we mean by porous media. So first, I just wanted to point out to you all that, heterogeneous and porous media are really quite ubiquitous. So for example, when you go outside and take a walk, right, you walk on soil and the ground beneath you, right, the ground beneath your feet is a heterogeneous and porous medium, right? Soils, sediments, even rocks that are way below the surface of the earth, right, which trap oil and gas and supply much of your drinking water, those are all heterogeneous porous media made up of grains of different minerals and solid materials that are all packed together, right? So here's actually one X-ray image of soil, right? And you can see that there's all this pore space in between. It's a very heterogeneous and disordered structure, not like the typical crystalline ordered structures that we learn about in our solid state physics, right? And so typically here, the grain sizes are anywhere between a micron and a millimeter in size, right? So these are pretty small pores, right? And these are important if we want to be able to understand and predict and control agricultural processes, oil and gas recovery, water remediation, and extract water from the ground and sequester carbon underground, right? So this is one ubiquitous example of a heterogeneous medium. I have to show this because I'm just, I just love showing this. This is a picture of my daughter, who is a little over a year old now. And as you can see here, she's incredibly adorable, but she's also incredibly, I guess she spits a lot. And you can see she's secreting mucus, right? And so mucus is actually a polymer solution. As Jenna pointed out in the discussion topic, your body, in fact, is made up of all sorts of heterogeneous media. For example, mucus in your lungs, right, is a heterogeneous porous medium, which is made up of mucin polymers that form a network, right? So here's an example of what this network looks like, right, in your lungs. And the structure of that network is important to clearing out pathogens and inhaled particulates and preventing bad stuff from getting to the underlying epithelial tissue, right? So that's an example of an important biological barrier in your body that is a heterogeneous and porous medium. Another example is in your gut. So your stomach is lined with mucus, your small intestine is lined with mucus, your colon is lined with mucus, and in fact, mucus in the gut is, again, a polymer network made up of mucin polymers that are often cross-linked, right? That forms a network. So here's a scanning electron microscopy image where the pores are anywhere between tens of nanometers in size to tens of microns in size, right? So that's another example of an important biological barrier whose function is to prevent bad stuff from getting through and reaching the underlying tissue. Your own tissues are heterogeneous media. So your tissues are made up of mammalian cells that come together and migrate in an extracellular matrix. And here you can see the scale bar shows an idea of the size of the pores in this heterogeneous medium, they're typically on the order of microns or up to tens of microns in size, right? So you are, in fact, a heterogeneous and porous medium. And, right, so Aiko, if she's good about brushing her teeth, but if she weren't good about brushing her teeth, she would get plaque on her teeth. You hear about this all the time. And in fact, just in general, your mouth has all sorts of bacterial communities, which are often known as biofilms, right? This is an image of actual biofilms that are in your mouth, right, and on your teeth. And those are also heterogeneous media. Last week, Andre Koshmirel told you about biofilms a bit, and he told you about how biofilms are collections of bacteria that come together and are encased in a polymer network, right, known as EPS. And this is a scanning electron microscope image of that, showing that, again, this is a heterogeneous and porous medium where the pores are micrometers in size. So the punchline here, right, is that heterogeneous and porous media are really ubiquitous, right? You encounter them when you go outside, under the surface of the earth, but also your entire body is made up of different kinds of heterogeneous and porous media. And that's what we study. And today's question that I'll present to you is what we've been focusing on is trying to understand how do bacteria move through these heterogeneous media. And it's important to note that in many of these cases, bacteria live in heterogeneous media where the pores are quite small. The pores are between 1 and 10 microns in size. And just to give you a sense of scale, right, the cell body length of E. coli, as you guys know from the reading, is around 2 microns long. So these are highly, highly confining structures. So these are tight media. And that's what we've been focusing on in our research. And so why do we care? Well, this is a fundamentally interesting physics problem in the physics of active matter, right? There's a lot of interest in just understanding the new physics that emerges when you have systems comprised of constituents that are actively taking in energy and moving around and propelling themselves, right? And bacteria are a canonical model system of active particles with tunable and diverse motility patterns. Right? But this is also important in many applications. For example, in agriculture, microbes interact with plant roots and help sustain a plant life, right? So we need to be able to predict and control how microbes interact with plants and move through soil to make that happen. In environmental applications, right, one very exciting area of environmental science is in using microbes to clean up contaminants. So for example, contaminated soils and sediments can often be cleaned up using microbes that move towards and degrade contaminants that are trapped. This is also important in health, right? As you know, many infections are microbial in nature, right? And to infect you, microbes have to burrow through the pores in your biological gels and tissues, right, that make you up. And so we need to be able to understand how they move through these heterogeneous porous media. And also, if we want to use genetically engineered microbes to deliver drugs to areas of the body. For example, one exciting area of research is in using genetically engineered bacteria to actually deliver drugs into tumors where it's hard to penetrate otherwise, right? And again, we need to be able to understand how microbes move through these heterogeneous porous media. So these are some of the reasons why we do the work that we do. And so again, I just wanted to highlight the people who make this work possible, right? Today, I'll tell you about some of the work that's coming out of my lab, right? We've been doing this for several years now. And this work has been really led by these 3 people on the screen. Tapa Bhattacharjee is the person who started this work in my lab. He's an incredible postdoc who joined 2 years ago and has been doing amazing work. And he led all the experiments that I'll tell you about. Daniel Amchin has been leading our theory. He's an incredible graduate student who has been leading all the theory and simulations that I'll tell you about. And Jen Ott is a new graduate student who's been working with Tapa and Daniel on really pushing this work forward. And we benefit tremendously from interactions with colleagues both at Princeton and elsewhere. We get a lot out of those interactions, and hopefully we'll also interact with many of you. So I want to start off with the discovery of bacteria, right? So this goes back to over 300 years ago by van Leeuwenhoek, who was a microscopist, a Dutch microscopist primarily. And so Leeuwenhoek invented this cool microscope, and he took a drop of pond water and he looked at it under the microscope, and this is what he reported. He said this was for him, among all the marvels that he's discovered in nature, the most marvelous of all, right? He saw many thousands of living creatures, all alive in a little drop of water, moving among one another, right? So this was the first study of microbial motility. He drew this picture of these bugs moving around, right? These were actually a form of microbes, as you read in the Howard Berg reading that we assigned before this lecture, right? So these were microbes moving around. So even from their very discovery, right, the ability of microbes, of bacteria to move, right, and swim through liquids has been a defining feature of a defining characteristic. He says, you know, there are 100,000 animalcules, he called them animalcules, in one small drop of water, right? So this was the first study of microbial motility. I find this paper that he wrote, or this letter, very amusing. Actually, if you keep reading it, Van Leeuwenhoek says, my method for seeing the smallest animalcules and the little eels, that's what he called these, I do not impart to others, nor yet that for seeing very many at once, but I keep that for myself alone. So this was, unfortunately, one of the first examples of what I would call an incomplete method section in a paper. Regardless, this observation has been replicated many, many, many times since, right? And as you read in the reading by Howard Berg, right, there's been beautiful work over the past few decades where people have really characterized in exquisite detail the ability of bacteria to move through different kinds of liquid environments. Right, and so this is a movie from Howard Berg's lab. Howard Berg did some of the seminal work studying E. coli, which is kind of a canonical example of flagellated bacteria moving. And with the case of E. coli, which have multiple flagella, right, they have these little appendages that you learned about that are flexible, that rotate, right, in one direction, and they bundle together, and they form what's known as a flagellar bundle. Right, so they're rotating, and that motion pushes the cell body along what's known as a run, right? So that's a constant speed motion along a straight path that for our E. coli, under our conditions, lasts around 2 seconds. So the flagella are rotating synchronously, they're pushing the cell body along, it moves along at a constant speed, and then after 2 seconds, one or more of these corkscrew appendages goes in the wrong direction. Right? And that causes all of the flagella to splay out, which causes the cell body to randomly reorient itself due to the hydrodynamic force. And then very quickly, the flagella rebundle into a coherent flagellar bundle. And then the cell moves along in another run at a constant speed, but in a different direction. Right? And this tumble process lasts around 100 milliseconds. Right? So much, much shorter than the duration of the run. So compared to a run, tumbles are effectively instantaneous. And so you can see these flagellar bundles in the movie here, and you can also see the tumbling where the flagella come apart, and then the cell moves off in a different direction. So this process is known as run and tumble motility, right? And so there were two really great questions that you guys submitted. One was, is there any way to predict movement accurately, or is it more of a follow and map approach, right? And Tapa addressed that a little bit in the discussion section earlier. Someone else asked, how does one know that it's better to model a process as a stochastic process rather than as a deterministic one? What are the pros and cons of both approaches, right? And so the way I think about this is kind of like how we model gas particles just randomly moving around, right? So in the room that you're sitting in right now, right, you have tons of gas particles that are just whizzing around and doing their thing, right? And they're bouncing off of each other randomly. And so in principle, one should be able to deterministically say, knowing all the positions and speeds, velocities of all the other particles in the room, I should be able to predict how one particle moves, but that maybe is not so useful in determining quantities that you want to know, like the pressure or how much something spreads out on average, right? And so for that, we usually go to stochastic models, right? And this is one example where Robert Brown basically dispersed pollen grains in water and watched them jiggling around, right? And that's known as Brownian motion, right? And the way we think about Brownian motion, right, it was put forward by Einstein actually in 1905, where he proposed that if you have a pollen grain in water, so that's this green thing, right? The water molecules are all randomly jiggling around. And so they push on the particle and they push it around in random directions, right? And that's why it randomly jiggles around. And so that was a theoretical explanation for Brownian motion, which was actually confirmed experimentally by Jean-Baptiste Perrin in 1911. And that was part of why he won the Nobel Prize in physics in 1926. So that's just a fun historical tidbit. But the point is, the way we model this process, right, is that this particle is randomly getting kicked around, right? So this is a random walk. And the way we characterize a random walk, right, on average, if there's no bias, this particle on average doesn't really move anywhere, right? Its mean displacement, right, is zero because it's just randomly moving in all directions. But you can characterize what's known as the mean square displacement, which is given by the position at a given time minus its starting point squared, right? And the angular brackets here refer to an average, which is an average over all the time steps. So for example, if I start here and I look at all times after that, right, I can do that for different starting points and average that. I can also average this over all the different particles if they're all behaving the same way, right? So this is known as an ensemble average. And the point is that someone asked, why is a quadratically varying MSD, or mean square displacement, an indication of ballistic motion? The point is, in general, for a random walk, right, you can see that if it has a characteristic step size, right, the step lengths are all distributed, right, according to some distribution, and the step speeds also have some distribution. But if you have a characteristic step length and step speed, then for times shorter than the characteristic step duration, right, this particle is just moving in at a constant velocity, right? It's not being hit pushed by another particle. And so the mean square displacement scales as time squared, because the mean square displacement is a length squared. So if you have constant speed motion, then the mean square displacement scales as t squared, right? And the slope of that gives you the speed, essentially, right? And so that's at short times, that's ballistic motion. But as you go to larger time scales, where now you get to see the randomness of this random walk, you transition to diffusive scaling, where the mean square displacement scales as t to the 1 power. And in fact, the prefactor gives you what's known as the diffusion coefficient, d, or the diffusivity. And that's an incredibly important parameter in describing how much this thing has spread out at a given time, right? So if you have a randomly moving object, right, it's very difficult to predict exactly what its trajectory is going to be. But on average, we can say after a certain amount of time has elapsed, it's spread out by so much from its original position. And that's given by the diffusion coefficient, which is set by the step length, L, and the step speed, V. So we can look at these E. coli, right? And this was a kind of a beautiful insight from Howard Berg, that if you look at this run and tumble motility, the runs have a characteristic speed and a characteristic length, right? And these tumbles are effectively uncorrelated and effectively instantaneous. And so on long time scales, this looks like a random walk. And it looks like a random walk where the diffusion coefficient is set again by the run speed times the run length, right? So this is a beautiful way of predicting how E. coli and many other flagellated bacteria and run and tumble particles in general, right, spread out in a homogeneous environment, right? That's what the diffusion coefficient parametrizes. And so this is what people have been looking at, right? And for over 300 years, typically we've studied bacteria in liquid, right? In just liquid cultures and test tubes, or on flat surfaces, right? But in reality, as I told you about, most bacteria actually live in tight heterogeneous media, whether they're in your body or outside in soils and sediments and subsurface rocks, right? They inhabit tight heterogeneous media. And the question that we've been addressing in my lab is, how is this beautiful picture of E. coli motion, right, and bacterial motion in general, how is it altered when the bacteria live in tight heterogeneous spaces? And so today, I'll tell you about our work looking at single cells moving, as well as collections of dense populations of cells moving via chemotaxis. But first, I just want to take a break and check the Q&A box to see if there's any questions that I can answer. So let me see how I can do this. Lana.
Sorry, Suji, I've been answering all the Q&A so far.
Oh, don't be sorry at all. That's what happens when you have amazing students. Okay, well, thank you. So there's nothing I need to answer then? Jenna?
Looks like there's some more questions coming in, so maybe if you want to check those since people are sending them now.
Okay, for some reason, I'm like incapable of operating technology, so I can't open up this Q&A box. Jenna, do you mind just reading out the question for me?
Yeah, totally. So I'm going to click the answer live on them as we answer them. So the first question is, if a run and tumble cell is transferred to a porous media, is there a lag before which the hopping starts?
Ooh, great question. Stay tuned, and hopefully I'll answer that in the next part of my talk. But
cool.
Yeah, we'll see. Any other questions?
Yes. So the next question is bacteria motion through a heterogeneous media chaotic?
Great question as well. It depends on how you define chaotic, but hopefully I will answer that as well. It is certainly random, and we will get to that very soon as well.
Cool. Next question is, bacteria behaves differently in vivo. We always say E. coli can divide every 20 minutes, but normally external forces are much more dominant, and of course this rate will drop.
Yes.
That's the end of it.
Okay, that's a great point. And I think that really highlights the importance of introducing different forms of complexity into the work that we do, right? So on one end, we can look at super idealized conditions where E. coli are dividing every 20 minutes, they're in a liquid culture, right? But you're completely right that in vivo, right, the conditions are very different. And so that's exactly what we're trying to do in our research, is bridge the gap between highly idealized lab experiments, which are incredibly powerful and tell us a lot about how these microbes function. But what we're trying to do is bridge the gap between that idealized picture and the full complexity of in vivo environments. And so I completely agree. The influence of mechanical forces, the influence of nutrient deprivation and different nutrient conditions, and heterogeneities, these are all things that we're actively working
on. Awesome. So there are a couple more, and I'm just trying to pick ones that I can't answer.
Yeah.
One second, Sujit, sorry.
No worries. And you know what? I have another Q&A break a little later on. So why don't I keep moving ahead, and then we can reconvene at that next Q&A break. Does that sound good?
Yeah, and I'll answer the ones that I can answer.
Cool. And Jenna, just while I have you, is my video okay and you guys can hear me okay and stuff?
Yep, everything's working great for me.
All right. Well, thank you all for the excellent questions. Keep them coming. First, I'll tell you about our work on single cells. So I gave you this reading from our Nature Communications paper, right? We also have a follow-up soft matter paper. And what we've been doing is looking at how single cells, exactly the question that you guys asked, how do bacteria move in heterogeneous media, right? So this is the run and tumble picture. Before we came along, we couldn't really find a good answer to the question. However, there are models that people have constructed. One very appealing model is to say that, well, in heterogeneous media, they still perform run and tumble motion, but now because they're in this heterogeneous matrix, right, they bounce off the walls. They collide with the walls of the solid matrix, and so their runs are shorter, right? So here, this beautiful paper says it best, where they say this model quantifies the intuition that in a dense matrix, bacteria are reoriented by collisions with the matrix as well as by just their intrinsic tumbles. The assumption is often made that the run speed is still unaltered. So within a heterogeneous medium, you can still calculate the diffusivity because they're still doing run and tumble motion. Now it's the run speed times the run length, which is shorter and given by something that's of order the pore size, right? And so that's something that you can characterize. The problem, right? So this is, again, like an analogy to gas diffusion. Gas diffusing inside a porous matrix, right? The molecules bounce off the walls and their diffusivity is hindered. The problem is that the predictions don't always agree with experimental results. So for example, if you take a heterogeneous or porous medium and put a suspension of swimming bacteria at one end and measure how long it takes for them to swim through the pores and reach the other end, use those measurements to extract a diffusivity and compare that to this estimate, the experiments are often lower by 1 to 2 orders of magnitude, right? And so we're clearly missing something. And this is the puzzle that we set out to resolve, right? What's going on in heterogeneous media? So how do you even study how bacteria move in heterogeneous media? Well, there's one very classic way of studying motility in microbiology, which is using what are known as semi-solid agar assays. Right? Semi-solid agar is like a goopy, viscous liquid, which is basically agar that's very weakly cross-linked, very low concentration. And then what you typically do is you take a plate, you put in a bolus of cells, and you watch them spread through the agar, right? So here are just some examples of that. However, there are some limitations, right? One is that the pore structure of semi-solid agar is very poorly defined. So if we want to be able to connect directly the heterogeneous structure of the pore space to how the bacteria move, it's very challenging to do that in semi-solid agar. And also, it's a messy system. It's hard to track individual cells for long periods of time and in 3D. Typically, right, you get surface effects because you have the top and bottom surfaces where oxygen can come in and the cells can go up to the surface. And it's very difficult, as you'll see later on, it's very difficult to control the structure of the overall community. And that's something that we've been able to overcome. And so what Datta did is he came up with a new approach, right? This is a platform that we use in my lab to study how bacteria move in porous media. And the idea is very simple. This is like a ball pit for cells, right? So what Datta does is he takes biocompatible hydrogel particles. So hydrogels are cross-linked networks of water-loving polymers, right? So they're crossing together. Datta dries them out and then redisperses them in whatever liquid culture medium that the cells like, right? And so the hydrogels, they swell up in the liquid medium, right? And they form these cross-linked balls, right? These hydrogel spheres, which are between 5 and 10 microns in diameter. But importantly, the mesh size, so the spacing between the links between the polymers within each particle is between 40 and 100 nanometers. And that's something we've measured. And so that's actually really big. That's big enough so that nutrients and oxygen and other small molecules can diffuse through the matrix completely unimpeded, right? But for the cells, this is a solid surface. And the cells have to move through the pores formed in between the particles. And we can tune the pore sizes by tuning either the particle size or how jammed together these particles are. If Datta disperses more solid dried hydrogel powder into the liquid medium, they swell up and they form a tighter packing. And so that way you can tune how big these pores are. And most importantly, this entire packing is transparent because the hydrogels are so swollen that it's mainly water, right? There's less than 1% of solid material here. And so you can see through everything. And we use what's confocal microscopy. So Josh, in one of his early lectures, talked about different forms of microscopy. We use confocal microscopy to actually look at what the cells are doing inside the pore space. So these are fluorescent cells. They're expressing a fluorescent protein in their cytoplasm. And so we use a strain of E. coli. And so this approach, right, enables us to isolate the influence of geometric heterogeneity, right? The fact that these particles are impeding how the cells move, right? But it doesn't impede how nutrients move. In real heterogeneous media, for example, in soil, right, you might have the additional feature that these particles also impede how nutrient moves, right? So that changes two things. And so we're starting to isolate the different influences of these different effects in our work. And so here's a map of the pore space structure. So TAPA dispersed fluorescent particles that move through the pore space, and you can map out the structure of the pore space. You can see it's very disordered. You can see there are these little channels around the particles, right? So that's the red is the pore space. And we can use these measurements to actually measure what the pore size distribution is in these porous media. And so this is a pore size distribution, right? So this plot shows you the fraction of pores that are smaller than a given size. So, so here, for example, this blue curve is for the loosest packing, where TAPA has dispersed the least amount of dry hydrogel powder, and there the pores are between 1 micron and 13 microns in size. And then as you add more and more solid powder, the pores get tighter and tighter as the particles pack together more and more. And at this point, this black curve shows you a porous medium where the pores are between 1 and 4 microns in size, right? And these are exponential distributions, characteristic of many heterogeneous and disordered media. And so that's the porous medium, that's a platform, and this is what we use to actually spy on how cells move inside the pore space. And so here's the punchline. So these are some of the first movies that TAPA showed me of E. coli, so that's the green stuff, moving through the pore space. So here we haven't labeled the pore space. The E. coli are moving through. These are just two separate cells. When TAPA showed me these movies, I completely freaked out. I fell out of my chair, right? And the reason is because, well, A, these movies are beautiful, but B, these cells are clearly not performing run and tumble motion, right? I told you that runs happen, they're straight lines, straight line constant speed, and then the cell very quickly reorients and tumbles, moves off on another run. Tumbles are essentially instantaneous. Whereas here, you can see these cells, as they move, right, they get stuck. This cell gets stuck, it reorients its cell body. It's constantly trying to reorient its cell body until it finds the right way out, until it moves again, until it gets stuck again, right? You can see they're constantly getting stuck. So we call this process hopping and trapping, right? The cell gets stuck there, it's trapped, and then it hops out until it gets trapped again, and so on and so forth, right? So these are two separate cells. You guys had some really great questions that I'm going to mention right now, but I just want to point out that this is a completely different mode of motility than run and tumble motion, right? And so this really blew our minds. You guys asked, is there a pattern of pores that locks motile cells in a regular, predictable pattern of movement, like a loop or a back and forth movement? I thought that was such a cool question. That's something that we'll think more about actually designing. But I just want to point out, look at this pore soil over here. This cell is stuck there, hops to there, gets stuck, and then this cell just constantly hops between the same traps over and over again. So even though this is a disordered porous medium, we didn't place these pores in any particular way. You can see that these kind of loops that force these cells to just move around in the same pattern do arise naturally. So this is a really interesting question, and we actually see that in our data. It seems necessary to have a flagellar assembly to hop in 3D environments. Are non-flagellated systems indefinitely trapped in a pore? Great question. So TAPA has tested mutants that don't assemble flagella, and indeed they go nowhere, right? Brownian, just regular thermal diffusion is not sufficient to let it to move these through the pore space appreciably. And so this is really due to the active motion of the cell. And I just want to mention the reason why we call this hopping and trapping is because this actually arises in many other forms of passive transport where you don't have active constituents. For example, it arises for colloids in a dense suspension where they get stuck and then they hop until they get stuck again. This actually ends up being how water molecules behave at cell membranes. They get stuck in one spot and then they move to another spot. This is how tracers in a biopolymer network move. They get stuck, they hop, they get stuck again, right? And in fact, this is even how often charges, electrons, move through various kinds of amorphous conductors. So this is actually some classic work where people showed that the way electrons move through amorphous materials is often can be described as a process of getting stuck in energy wells and then hopping out of them and getting stuck in another energy well, and so on and so forth. So this is actually where the term hopping and trapping started, and so we called our bacteria motility hopping and trapping by analogy to these other forms of transport. So there appears to be this really interesting connection with transport in other systems. And so we can look at the mean square displacement again, right? In free media, as I mentioned before, at short times, the cells move ballistically, and then at long times, they move diffusively because they're performing run and tumble motion. In a porous and heterogeneous medium, right, we would think that maybe this crossover point would simply get shifted to earlier times, but instead, what happens is it gets shifted, but also the scalings of the mean square displacements become subdiffusive, right? We can rescale these by the crossover point, and indeed we see that for all of these different porous media. So here are the pore sizes. At short times, the cells move super diffusively, and then they transition to subdiffusive motion, right? Which is a hallmark of this transient trapping. And as you make the pores tighter and tighter, they become more and more subdiffusive, and at longer times, they eventually become diffusive, right? So this MSD, again, reflects what we're seeing in these movies, where this trapping is a transient process. As they explore through the pore space, they get stuck transiently before they keep moving. So we can separate out traps and hops by tracking the individual cells. For example, the instantaneous speed is low when a cell is stuck, and then it is high when it hops through the pore space. Tapa can look at how the orientation moves, how the orientation changes. So this is how the velocity, orientation of the velocity vectors change as the cells move through the pore space. When it's pi, that means the cell is just moving back and forth. It's rattling around. When it's 0, that means it's moving in a straight line, right? When the cell is trapped, this angle is all over the map. The cell is just randomly trying to get out. When it's hopping, it's 0. The cell is moving in a directed path through the pore space. You can see that again here. This is the distribution of these angles, where for trapped cells, right, this angle is all over the place. For hopping cells, you see this strong peak near 0. So hops are directed paths through the pore space. Traps, the cell is stuck and constantly trying to get out. And so one natural question is, what determines the hopping length? I'm going to skip over the details of this because Tapa talked about this in the discussion earlier today, but essentially hops are truncated runs. The cell is trying to run, but it hits a wall, right? And so we can actually predict the distribution of the hopping lengths. So that's what you see here, right? By the distribution of straight line paths in the pore space. And so this is known as a chord length distribution function, which was actually described by my colleague Sal Torquato here in chemistry in the 90s, right? So we used this chord length distribution function, and it works at predicting what the hop lengths are, right? And as you make the pores tighter and tighter, the hop lengths get smaller and smaller, right? This is the distribution of hopping orientations. You can see it's pretty much distributed all over the place because it's a random porous medium. So the cells hop in all directions. We can also label the flagella. So Tapa labeled the flagella here in magenta. And I think this movie is so incredible. I'm going to play it again, right? I mentioned that every 2 seconds in liquid media, the cell is trying to unbundle its flagella and rebundle it, right? But you can see here the flagella remain bundled for up to 11 seconds before the cell finds the right orientation or one of the flagella to unbundle. And then that enables the rest of the flagella to rebundle in a different position, and that lets the cell move out, right? So the flagella remain bundled for longer periods of time because they're stuck in this tight spot. But then when the cell finds the right orientation for one of the flagella to unbundle, then they all unbundle and rebundle in a different spot, and that enables the cell to move out in a different direction. So it looks like trapping is related to the fact that confinement suppresses unbundling, and then when one of the flagella can unbundle and the flagella rebundle, that enables the cell to continue to move through the pore space. And so how do we model this? So Tapa talked about this in the discussion section earlier, but we can actually draw an analogy to polymers trapped in porous media, which are randomly trying to get out of tight spots due to KT, thermal energy, and using a model inspired by models of polymer moving through porous media, what we can actually predict is that we expect that the distribution of trapping durations should scale as a power law, where the exponent quantifies the competition between how actively the cell is trying to get out and move through the pore space versus how tightly confined the cell is. And indeed, that's what we see in the data. We see these power law tails where the exponent depends on how tight the pores are, or by tuning the temperature, we can actually tune how actively the cells are moving through the pore space, and that modulates the power law exponent as well, right? So trapping is set by the competition between cell activity and confinement in the pore space. And so long story short, we have a revised view of how single cells move through a porous medium, right? We don't have this picture of run and tumble where the runs are now truncated, but instead we have a different mode of motility where the cells hop and then get trapped for long periods of time and then hop again. Hopping lengths are set by the geometry of the pore space, whereas trapping durations are set by the geometry of the pore space and how active the cell is, right? And these are all quantities that we can measure and predict, so we can put them together, and this still looks like a random walk. It's just a weird kind of random walk, where now the diffusivity is set primarily by the hop length squared divided by the trapping duration, because the traps are the larger time scale. So we can put these quantities together and we can come up with a new prediction for what the diffusivity of the cells should be. And we can test that. So Tapa takes a dilute population of cells, he embeds it in a porous medium, and he watches over a very long time scale how the leading edge of this spreads out due to motility. So these are just 3 different measurements in 3 different porous media. The line, so actually the y-axis here is the measurement. The x-axis is the prediction from this hopping and trapping model, just using the mean hopping length and the mean trapping time, right? Which we know is not perfect because we know there are these long distributions, but actually, as this line shows, it works pretty well to within a factor of 3. We thought this was incredible because just as a point of comparison, if you take the old run and tumble model and just replace the runs by the pore size, the prediction that model makes is off of your screen. It's way over here. You can't see where my hand is, but it's way, way off your screen. So we can do much better in predicting the diffusivity of these cells using this hopping and trapping model. So this gives you an idea of how single cell motility is altered in heterogeneous media. I'm going to take another pause for a Q&A break, and then really quickly, I'll tell you about the work we've been doing looking at collective behavior in more dense populations. So actually, Jenna, if you don't mind, or Daniel or Tapa, can you guys tell me about some of the more pressing and salient questions that have arisen?
Yes, definitely. So one of the questions is, have there been any interesting connections to glassy dynamics, example, cooperative rearranging regions?
That's a great point. So these cooperatively rearranging regions are like strings of particle motions that are observed in glasses and supercooled fluids. I have to think about that a little bit. The main connection to glassy dynamics is actually through this, how we understand these trapping durations. There's actually a very beautiful connection to glassy dynamics that we found that we talk about in this soft matter paper. So I encourage you to take a look at that if you're interested in it. And then about the cooperative, the CRRs, I have to think about that. That's a very interesting question.
Cool. All right. So next question is, what is the difference in the assumption between the collision-based heterogeneous motility model and Drude's theory of electron motion in conductors?
Oh, wow. That's a really interesting question. Um, I think um, I don't know if I can answer that succinctly in the time that we have. So let me think about that. And whoever asked that question, if you want to email me, I'd be happy to follow up afterwards.
Awesome. All right. Next question. What are some of the ways we idealize these systems, just as we idealize systems involving gas particles if it's analogous to gas diffusion?
Right. So, the punchline here is that it's not perfectly analogous to gas diffusion. What we found is it's kind of like gas motion where like the gas sticks to the walls in a sense. And there are different forms of gas diffusion. Actually, there's Knudsen diffusion and whatnot. But I think the question was, what are the main idealizations that we make? Um, the main idealization is essentially in this picture where we're saying that the diffusivity is set primarily by the hop length and the trapping duration. There's also, by the way, a hopping duration, which we can incorporate to be even more precise. Yeah, I'm not sure how to answer that question. Let me think. Whoever asked that, if you can follow up with me and elaborate on what you mean, I can follow up with you.
Cool. And another one is, can't hopping and trapping be considered as a run and tumble, with a trapping being a run cut short followed by tumble before another run is allowed?
So no, and that's the interesting part is that not really, because as we found, right, the trapping duration. So let me put it here. The trapping durations can be, up to 40 seconds, whereas the typical tumble time, like the typical run duration, is only 2 seconds in the liquid media, right? And what we see here, right, is that this cell gets trapped for a long period of time, and then at some point it finds the right orientation for the flagella to unbundle and rebundle. So it looks like there's this interesting interplay between the flagellar properties, the pore geometry, and the surface interactions that determines the duration of trapping. And so I don't think it's as simple as just saying that it's run and tumble, but truncated in some way. I hope I answered that question.
Cool. And then maybe one more before you go
Perfect.
again. When an electron gets stuck in a well, it releases energy and then absorbs energy while moving up. Is it applicable for bacteria as well? What role does the heterogeneous media play in this energy interaction?
That's a really interesting question. I think that is more specific to just electron behavior. Here, the bacteria aren't, they're not like jumping to an excited state or anything like that. Um, I don't think those effects arise in any way in the bacterial system, but it's an interesting question. The question about what's the role of the heterogeneity is essentially in this landscape that's very disordered of different energy well depths that the cells navigate as they move through the heterogeneous medium. And the way that manifests is as a distribution of essentially confinements or well depths. And that gives rise to this power law distribution of trapping durations. It's very interesting. This ends up being ubiquitous in many disordered systems. And if you're interested in that, check out this paper, which is like super in depth, but also check out our soft matter paper if you want to learn more about that analogy. And I guess with that, I'll keep moving, and I'll end by just telling you about our work on denser populations, since there's only 5 or so minutes left. I'll just give you a brief glimpse of some of our newest work looking at dense populations, and then we can pause there. So I'll tell you about chemotaxis. And so this work is really inspired by a classic paper by Adler in the 1960s that I encourage you all to read. What Adler did is he took E. coli and he just put a dense plug of them at one end of a glass capillary. So this is a glass tube filled with liquid media, right, with multiple different nutrient sources for the bacteria. For example, aspartate and serine, 2 amino acids. He put a plug of E. coli at the end, and what he found is that the cells actually formed these traveling wave fronts, right? So this is a cluster of cells that's moving from left to right through this capillary, followed by another one moving, right? And the reason why is because they're actually consuming nutrients, right? So for example, here they're consuming serine, and they're setting up a gradient that they bias their motion towards. This is what chemotaxis is, right? And so someone asked this question, do E. coli only detect aspartate? Or what other chemicals do E. coli detect? I just mentioned serine, that's another one. There is a whole plethora of chemical signals that E. coli bias their motion towards. And if you follow up with me, I can tell you more about that. But this is a classic experiment that showed the formation of these traveling fronts of cells that coherently migrate. People have since done beautiful microfluidic experiments also showing how these things travel as these bands through channels. More recently, people have looked at this in semi-solid agar and shown that the cells respond to different kinds of chemical cues and nutrients in different ways. And this is how they kind of spread out, explore new terrain. And so why are they doing this? Here's a simple picture that was developed by Keller and Segel in the 70s. And it's a really beautiful model known as the Keller-Segel model. Imagine you have a collection of dense bacteria in green, right? So B, and they're surrounded, let's say, by a nutrient or some chemical signal that they metabolize, right? So the nutrient is diffusing in from everywhere, and the cells are eating the nutrient. So what happens is they deplete the nutrient. Okay, so you can model this, right? So let's skip these equations for a second. They deplete the nutrient locally, right? And what happens is they sense this gradient of nutrient. They sense that they've depleted nutrients locally. And so as they spread out and move, they bias their motion towards higher nutrient levels, right? They bias their motion up this nutrient gradient, right? So bacteria are like people. We sit here, we deplete our nutrients, right, on this planet, and then what do? We try to go somewhere else. We try to go to Mars or something and get more nutrients. So these cells do the same thing. They deplete their nutrients. They sense that there's a gradient of nutrients. They bias their motion outward. They continue to deplete nutrients as they move. They continue to consume nutrients, and they propagate that nutrient gradient and continue to bias their motion. And this gives rise to these bands, these propagating fronts of cells that move coherently, right? So the Keller-Segel model quantifies this by considering the diffusivity of the cells, chemotaxis, which is parameterized by this parameter, which is known as the chemotactic sensitivity coefficient, that quantifies how sensitive the cells are to this nutrient gradient, and growth. And you couple this equation for the bacterial dynamics to an equation for the nutrient dynamics, where the nutrient diffuses and it's consumed by the cells. And this model gives rise to these traveling fronts of cells. And so we ask the question again, this is all well and good. People have studied this in homogeneous media, right, on flat plates, in viscous liquids, in microfluidic channels. What about in heterogeneous media, right? And so this is where another important feature of our experimental platform comes into play. Tapa figured out a way to 3D print in these porous media, in these heterogeneous media. I told you these things are ball pits for cells, right? So they're crammed together particles that are squished together, right? And so you can actually inject cells into the pore space. So this is a side view of Tapa injecting colloids into the pore space. And look, they just get stuck. You take a nozzle, you inject stuff into the pore space, you move the nozzle through, and the stuff just sits there, right? So we can 3D print, for example, the Princeton shield. These different colors are different layers at different depths, right? We can make these structures out of cells. So you can 3D print any microbial community of any structure and any composition that you want, right? So this is something we're very excited about, and we're pushing forward in many different directions. But to study chemotaxis, Tapa did the simplest possible thing. You just 3D printed a cylinder of cells embedded within a 3D porous medium, within this 3D medium. So this is a cut through them. This is way zoomed out. So each pixel here is a single cell. This length scale, the width of your screen, right, is millimeters, right? So it's thousands of cell body lengths. These are really large length scales. And here's what we see when we just watch this thing, right? I thought this would just spread out diffusively because the cells are randomly moving around. We see these propagating fronts. This movie is going to loop. Notice the time scale. These are hours, many, many cell divisions, right? You can see that the cells spread out and move coherently in these traveling fronts. And so we've been studying these. These are chemotactic fronts, right, that are moving throughout this heterogeneous pore space, right? So this is a top view. We can look at it from the side because these are 3D images, right? You can see that the cylinder is expanding outward throughout the pore space. You can zoom in. Oh, you can make the pores smaller. That changes how sharp these fronts are and how quickly they move, right? So that really shows that heterogeneity matters, right? You can, again, look at it from the side. You can see this cylinder expanding outward, right? And so we've been having a lot of fun exploring how the classic models of chemotaxis apply to cell motion in heterogeneous media. That it turns out the Keller-Segel model works. So Daniel did these simulations showing that the front surfs its nutrient gradient and it forms this peak that moves out. And he showed that you can capture, using the Keller-Segel model, the influence of confinement, but you have to update the transport parameters. You have to update the chemotactic sensitivity and the diffusivity using the hopping and trapping model. Someone asked, can we have hopping and chemotaxis simultaneously? That's exactly what these fronts are. When we zoom in, we can see that the cells are still hopping and trapping, right? But there's a bias and that lets them move out through the pore space. In the interest of time, I'm gonna skip the details of how the cells are chemotaxing. It turns out that the typical mechanism that Tapa talked about in the discussion section, that the cells, modulate the frequency with which they tumble, does not work in heterogeneous media. And it turns out that the cells use a fundamentally different mechanism to bias their motion by hopping more frequently up the gradient than down the gradient. So it turns out that heterogeneous confinement changes fundamentally how the cells chemotax, right? But they can still collectively move in these fronts. And then finally, I'm going to end with this fun thing here. We can draw all sorts of things. So Tapa and Jenna drew a squiggle of cells. And what you see here is that chemotaxis actually enables them to smooth out their initial morphology, right? So chemotaxis regulates the shape of a population, but if you make the pores too tight, then the cells can't move anymore. They're just stuck and they just grow and divide. And in that case, we see this abrupt transition to a roughening behavior, right? So this is another really beautiful set of physics and instabilities that we're starting to explore using these microbial communities. And so with that, I'm going to conclude. What I hope I've shown you is that in heterogeneous media, where you have strong confinement, bacteria move in a completely different way. We see this new mode of motility that we call hopping and trapping, which is controlled by the competition between confinement and how active the cells are, right? And by looking at this hopping and trapping model, you can actually predict the long-time diffusivity. And it turns out you can also quantify chemotaxis and collective migration via chemotaxis throughout these heterogeneous media. And finally, we've seen that pore size and how tightly confined the cells are regulates the morphology of the overall community. It lets them either smooth out perturbations or it amplifies perturbations and causes the community to roughen in new and interesting ways. So with that, I just want to say this is the tip of the iceberg. You guys had some other questions for work that we're doing now and work that we're planning to do. For example, do they only detect aspartate? No. So I showed you these fronts where they're only responding to one chemical signal, but in more complex media with multiple signals, we've actually seen multiple fronts, just like Adler did. What are the limitations in comparing our platform to real porous media? I touched on this, right? This is not perfect, right? This is not a perfect representation of tissue or of soil, right? But what we've done is we've started to isolate the influence of geometric confinement, right? Because you have these obstacles in the way, but there are also chemical heterogeneities that arise in natural media that we're starting to put into our hydrogels. There are structural heterogeneity. Someone asked a question about pore size gradients. I thought it was amazing. That's something we're trying out. Nutrient heterogeneities. These are all things that we're starting to incorporate in the experimental platform. What are the uses in everyday life? How do we influence this? Our hope is that this new understanding that we have can help us develop more accurate models to predict and control how bacteria move in agriculture, in tissues, in drug delivery, in bioremediation. So this is just the tip of the iceberg. Can we do this with other bacteria? Indeed, we can. We have. We are continuing to. Other bacteria have other motility patterns, and that manifests in interesting ways in heterogeneous media. Can they be recovered? We think it's very exciting. We can actually take them out, do other sorts of assays. Stay tuned. We're very excited about this. And with this experimental platform, there are many different directions that we're heading. So with that, I wanted to thank you all for listening, for your amazing questions. I'm going to stick around for a few more minutes to see if there are any other questions. This is my incredible group. They do all the work that I told you about. And thank you all. And I'd love to hear any questions you have. You can check out our website here. You can check out our Twitter, which my students run. If you have any other questions, email me. I'd be happy to chat more. So with that, I'm going to stop and take any other questions you guys have.
Do you want me to do some of the Q&A that's left over?
That would be awesome.
Cool. So one of the questions right now is, what is the mean loss in speed and energy when a single bacterium changes its direction? Has it been studied yet?
That's an interesting question. I don't know. So the short answer is I don't think there is any loss. Yeah, I think that's a pretty complex question, actually.
Cool. Next question is, how did you define these epochs of hopping and trapping for the movement change in direction angle time series?
Great question. So basically, we set a speed threshold. So if the cells are not moving, or if they're moving too slowly, they're trapped. If they're moving quick enough, then they're hopping, right? And that's something where we varied the threshold and seeing that the results of how we separate out hops and traps are relatively insensitive to that specific threshold, but that's a good question. Basically, when they're moving or when they're not moving.
Cool. Next question is there a general equation for bacteria motion in porous materials? Does Keller-Segel model work only for gradient?
That's a great question. So it depends at what scale you're looking, right? If you're looking at large length scales, much larger than the individual cell length scale, then yes, you can use a diffusion equation in the absence of a gradient, right? And then with a gradient, the Keller-Segel model is basically the diffusion equation with the influence of a bias due to a gradient. So that's a great question.
Awesome. Next question says, Dr. Datta, regarding the 3D printing in porous media, do you see any real-world biomedical applications stemming from that?
Oh, tons. I mean, we can 3D print designer microbial communities. Microbial communities are super beneficial for all sorts of applications, for water remediation, for different kinds of healthcare applications in terms of, different kinds of healthcare applications, for studies of socio-microbiology. There are tons of applications that we're excited about and we're working on right now.
Yep. And last question, it looks like, is, are there equations to account for the effect of the heterogeneity of media on the motion specific to the composition of the media?
I'm not sure I fully understand the question, but if you're asking about chemical composition or the influence of chemical effects and chemical heterogeneities, we're still working on that. Stay tuned. So this is brand new work. I want to tell you guys about our latest work. We've just scratched the tip of the iceberg, and there's a lot more to do.
Yep, so I think in a lot of the questions I answered as well, it seems like they are reading our mind for the next directions for the lab. So definitely check out our lab website, because I think that will contain a lot of our next steps.
That's awesome. Thank you all for listening. If you have any more questions, email me, email Daniel, Jenna, Tapa, and this was a real pleasure. Okay. So Lana, I think we can end right there.